The next important thing is coenzymes. In your chapter, the last thing is coenzyme and isoenzyme and its applications. So another one important thing is coenzyme. So what is coenzyme? So the coenzyme is nothing but it's a non-protein organic low molecular weight substance. It is a non-protein substance. It's a organic substance, even inorganic also possible. So these are the non-protein low molecular weight substance. It's always associated with enzyme function. So we still now we know that the enzyme is going to catalyze the reaction. The enzyme is convert the substrate into product. So in that mechanism, we have additionally we must know that in that process, some of the non-protein substance, low molecular weight substance, it's also involved the enzyme function. Means that specific substance it's called as coenzyme. Still now we know that the enzymes is going to convert the substrate into product. Here at another one additional point, in that reaction some of the organic or inorganic otherwise non-protein substance it's involved the reaction, means that's called as coenzymes. Okay, so many enzymes are require such a non-protein small additional factors for carry out the reactions. So those factors is called as cofactors or coenzymes. Mostly the cofactors are organic or inorganic in nature. So it may be the organic or inorganic nature. So the based upon the things we have named as a enzymes in different format, you can see the functional enzymes is referred as a holoenzymes. So the holoenzyme means nothing but the enzyme is going to function. If any enzyme is going to work functionally, means it is called as holoenzymes. So in that subcategory, if the enzyme is alone, means it is called as aphoenzyme. The enzyme is alone. Alone means it doesn't having any inorganic substance, otherwise cofactors, nothing is there. Just it's just made up of protein or polypeptides. It's having such unique role, means it's an enzyme. So it doesn't have any cofactors, means it is called as aphoenzyme. That means it's without having any cofactors. So non-protein part, we know that along with the enzyme it's having some other protein non-protein substance, means the non-protein substance termed it's called as coenzyme. The protein part the same thing. If it is an enzyme, if it is alone, it doesn't having any kind of cofactors, means it's called as a aphoenzyme. So in along with the aphoenzyme, some of the activator is there, that means it's a cofactor, it's called as a coenzymes. So if any enzyme is going to functionally it's going to work, means it is called as holoenzyme. If it is alone, means aphoenzymes, along with the protein substance, means is coenzyme. Total is going to work out, means it's a holoenzyme. The term is a prosthetic group, means the non-protein moiety, that means it's a coenzyme, it's tightly bound with the enzyme which not easily separate even by dialysis. That means these are the coenzymes is going to tightly bond with aphoenzymes. The coenzyme means non-protein substance, aphoenzymes is plain enzymes. So this coenzyme is going to tightly bound with the aphoenzyme, means when it's going to functionally work, it's called as holoenzyme. So this coenzyme is going to tightly bound. No, that bond it's can't able to break even when you are going to like separating dialysis. Gel electrophoresis method, best example for that. Even you can try for the gel electrophoresis, that coenzymes it's not going to separate, it must be come along with the aphoenzymes. So the TAM activator is referred as a inorganic cofactors, example calcium, magnesium, manganese and all. So it's going to enhance the activity. So this coenzymes it's also is called as a second substrate. This coenzymes is also called as a second substrate because it's going to bind with the enzymes. After that substrate is going to bound, it's converted into the product. Why it's called as a second substrate or co-substrate means it's having a high affinity, the same like substrate, it's also is having high affinity with the enzymes. when compared to the substrate. How much the affinity is there? Now the same kind, otherwise more than substrate affinity, the coenzymes having affinity with the enzyme. So it is called as a co-substrate. So the coenzymes is undergone the alteration during enzymatic reactions. That means during during the enzymatic reactions, the coenzymes also it's undergone the alterations. That means is may going to donate something else, otherwise is going to get something else. So during during enzymatic reactions, this coenzymes also is going to undergone the alteration. You can see there, but it's a totally vice versa. It's contrast to the substrate, because the substrate it's converted into the product, but here the coenzyme it's not converted into the product. It's going to lose something else, otherwise it's going to donate something else. After that it's going to regain some the same thing. It's get back to the same position or same nature. But the substrate, once it's going to bind with the enzyme, it's definitely is going to convert it into the product. So the reaction, best example, the coenzyme is going to participate in various reactions. It's involved the transfer of either atoms or groups like hydrogen, aldehyde, keto group, amino group, acyl group, methyl, whatever it is. So it's going to donate, otherwise it's going to transfer the some specific atoms or group during the enzymatic reactions. So it's may donate. So the coenzyme is going to donate such a group of group, otherwise atoms, during enzymatic reactions. So the coenzymes is play important role, decisive role in enzyme function. Indirectly we can say the enzyme functions also. The responsible item is coenzymes. So the same thing, certain coenzymes is transfer the substrate from one point to another point in cell. That means inside the cell itself, the enzymes in along with the coenzyme, now the coenzyme is going to transport the substrate from the one point to another point in cell. So it's simply it's act as a shuttle, serve as a recyclable shuttle. It's like a transporter. The coenzyme UDP, it's combined. The example, the UDP is combines with the substrate glucose. It's going to form a UDP glucose. So the coenzyme is UDP. The substrate is glucose. It's going to form a UDP glucose. So during this inside the cell, the glucose is made transport from one point to another point in the form of UDP glucose format. So when the coenzyme is going to release, the UDP is going to release. That time the glucose is going to free. It's undergone the further process. So the coenzymes is also it act as a recyclable shuttles. So in all this case, the coenzyme is going to facilitate, coenzyme is going to facilitate the recognition of substrate by their target enzyme. That means it's going to helpful for, that means the coenzyme is it's also facilitate the recognition of substrate. That means if a target enzyme is there, the target enzyme it can't able to easily find out the substrate. So for those case, your coenzyme is going to act as a recognition unit. So next, what is exactly coenzymes? Where it's come? Where it actually? What is the chemical nature means? Mostly the coenzymes for D-complex vitamins. Mostly the coenzymes are D-complex vitamins. That means it's derived from the vitamins. So mostly the coenzymes are derivatives of water-soluble D-complex vitamins. So during biochemical reaction, otherwise the vitamins is act as a coenzyme during various biochemical functions. So these are the table. I have listed the various types of coenzymes where it's derived from. I can give some example. You can see that the coenzyme thymine pyrophosphate TPP. Actually, it is derived from the thymine. So what kind of group is going to transfer means it's an aldehyde group and keto group. So the dependent enzyme, the specific enzyme is transcalidase. So the transcalidase is an enzyme. It's going to transfer the aldehyde or keto. So in that reaction, the coenzyme is thymine pyrophosphate. That thymine pyrophosphate it's actually derived from thymine. So this is the statement. So the same manner, various enzymes. These are the list of the enzymes. The various enzymes It's carry out some unique functions with the help of these coenzymes. These coenzymes are exactly derived from this kind of amino acids. So this is the concept. So in last slide we said mostly it's a vitamins. So apart from vitamins, some of the non-vitamin substance also is act as a coenzymes. So non-vitamin coenzymes. Not all the coenzymes are vitamin derivatives. Some exception is there. Some of the organic substance, it's act as a coenzyme. It's no relation between the vitamins and all. So some of the organic substance, it's also act as a coenzyme. So they may be considered as a non-vitamin coenzymes. So it's considered as a non-vitamin coenzyme. The example ATP, CDP, and UDP. So we know that what is ATP and CDP and all. Adenosine, you can see this table. Adenosine triphosphate. The abbreviation is ADP. So it's also is going to involve the act as a coenzyme. So it's going to donate the phosphates. So we know that already we have discussed in the high energy rich compounds and all. So it's going to donate the phosphate group. So whenever it's going to phosphorylation, the phosphodiesterase bond is going to break, it's produce the energy. So apart from that, it's going to donate the phosphate group. Once it's going to donate the lose the phosphate group means it's converted into the ADP. Once again, it's going to donate means it's further is converted into the AMP, adenosine monophosphate. So this is the example. So it's going to donate the phosphate group. So it's act as a coenzyme category. So along with that, some nucleotide coenzymes also there. The examples, the nucleotides, you know that it's a combination of nitrogenous base, one sugar, and phosphate. These are the three things is going to combine means it's a nucleotides. The nucleotides is nothing but it is act as a base pair for the DNA chain. So DNA, you know that it's a chromosomes. Chromosomes are present on the nucleus for eukaryotic cells. So whatever it is, eukaryotic or prokaryotic, it's having a chromosomes. Chromosomes are made up of DNA. DNA is having made up of nucleotides. Nucleotides is nothing but it's a base pairs, various base pairs. So the nitrogenous compounds is nucleotides. For example, NAG plus, NAGP, and FAD, it's coenzyme. So adella vittunga. So you can see there some of the proteins, thioredoxin, it's a protein, it serves as a coenzyme. Thioredoxin, it's also protein substance, it's also is act as a coenzyme. So vitamins also is act as a coenzymes. Some organic non-vitamin substances also act as a coenzyme. Here nucleotides also is act as a coenzyme. The fourth one is proteins. So these are the four different para things is going to act as a coenzymes. So here another one myth is there. So still now we know that the coenzyme is involved the reactions, otherwise is going to facilitate the find the target enzyme, all those things. But the coenzymes do not decide the enzyme specificity. So in previous slides we have discussed some enzyme is going to act on the some specificity, stereospecificity and absolute facility. All those things we have discussed in last slide. But that kind of enzyme specificity, it's not going to decide, not going to alter by your coenzyme. That means your coenzyme, it's not at all going to decide the enzyme function. You can see there the particular coenzyme, it may participate in catalytic reactions along with different enzymes. That means the coenzyme, we know that it's going to facilitate the enzyme function. That's a statement. But that coenzymes, it's going to act on different enzymes also, but it's never going to decide the enzyme function. The example you can see there, NAD plus. So the NAD is act as a coenzyme. Okay, so it's going to donate the some groups as donate the some atoms during the enzymatic catalytic reactions. So you can see there the NAD plus is act as a coenzyme for two different enzymes, lactate dehydrogenase and algal dehydrogenase. So both the enzymes function is different. The lactate dehydrogenase is responsible for the conversion of lactate into pyruvate. That's a role of the lactate dehydrogenase enzyme. Algal dehydrogenase is responsible for the conversion of algal into acetaldehyde. That's a role. Alcohol is converted into the acetaldehyde. That's reaction catalyzed by alcohol dehydrogenase enzyme. So the two different enzymes carry out the two different unique function, but the coenzyme is unique. That means it's indicate your coenzyme is not going to decide what kind of reaction is going to happen for the specific enzyme. That means specificity, enzyme specificity, it's not depend upon the your coenzyme. That is uniqueness. The both the enzymatic reactions, the NAD plus, it's involved the hydrogen transfer. So it's involved the reactions, it's involved in the catalytic reactions as a coenzyme. Its duty is just is going to donate the hydrogen group. That means it's going to transfer the hydrogen group from one group to another product during the formation of substrate and product. Apart from that, it doesn't having any role for the enzyme actions. So the specificity of the enzyme is mostly depend upon the AFO enzyme. So AFO enzyme means we know that it's a pure enzyme without coenzyme. So the AFO enzymes is responsible for the enzyme specificity, not upon your coenzymes.